Yes, thank you very much for a very kind introduction. Good morning, everybody. It's my big honor to attend this virtual conference, and thank again for invitation. Uh, my today's talk will be about gradient copolymers based on two oxazolines and their utilization for photodynamic therapy. It is known that nowadays method for non-invasive treatment of cancer is in high demand, and one of them is also photodynamic therapy. This is a treatment which involves the light, and light source is using for destroy the abnormal cells. Today, it can be used for treatment of cancer, also for treatment of different skin diseases, and even during the COVID time, photodynamic therapy was used for sterilization of materials against microbes, and we know also antimicrobial photodynamic therapy. The principle behind photodynamic therapy is in the energy transfer, where the light source is exciting the photosynthesizer to produce single state, and then energy is transferred to the production of reactive oxygen molecules. We recognize two types. First type is a generation of reactive oxygen species like peroxides, and second type is based on the production of singlet oxygen. These highly reactive molecules can destroy the abnormal cells. Recently, the number of possible photosynthesizers is possible to use, mostly based on porphyrins and phthalocyanins. Here you can see one unique molecule called hypericin. Hypericin is a naturally occurred compound from the Sanjom plant, and now is a very powerful photosynthesizer found in nature. The advantage is that the absorption maximum is about 600 nanometers. It means the light is relatively low energy. What is very specific feature of hypericin that light is emitting only in monomeric form. It means when hypericin is aggregated, no fluorescence is occurred. Drawbacks of hypericin is in very low solubility in water. It means it's very hydrophobic, and it is not possible to distribute to physiological media. One of possibility is use the nanomedicine. It means the encapsulate of hypericin inside nanoparticles, and then deliver hypericin to cell to produce energy transfer and generation of the reactive oxygen species. In spite of the fact that hypericin is very effective photosynthesizer, only very few examples for encapsulation and delivery of hypericin through nanoparticles can be found in the literature. In the the study of Professor Mishkovsky, when I recently cooperate, they tested lipoproteins and lipoproteins covered by cyclodextrin to deliver hypericin, but they observed very fast redistribution of hypericin and very fast release would leads to not so efficient photodynamic activity. Second example was in the paper in Professor Han. He used block polymers of polyethylene glycol and polycaprolactone, but they observed very low drug loading capacity and low stability of the prepared nanoparticles. As you could hear yesterday, one of the excellent areas or types of polymers suitable for biomedical application are polyoxazolines. They can be used as building blocks for synthesis of different advanced materials, hydrogels, nanoparticles, and so on. And the main advantage is in the preparation via living cationic polymerization, when you can control our size, architecture, and you can introduce different functionalities as end groups or in the side chain. Altering substituents in side chain, you can very nicely control polarity of polymers from the highly water soluble to highly hydrophobic polymers. Such feature can be used for preparation of different amphiphilic materials. Principally, for preparation of nanoparticles, you can use two types of copolymers, which can provide self-assembly. First is block copolymers, providing traditionally two-step synthesis, and with distinctly separated hydrophilic and hydrophobic blocks. Second possibility are gradient copolymers, producing in one photosynthesis. They have gradual distribution of copolymers and without distinct border of lipophilic and hydrophilic parts. We recently published a paper where we showed different behavior of block and gradient copolymers. You can see from the Langmuir-Blodgett relationship that gradient copolymer produce monolayer with lower surface area and a higher surface pressure compared to block analogs. It means that in air-liquid interface, gradient copolymers behave as more rigid molecules. This is due to the distribution of rigid aromatic rings toward whole molecules. This difference in the aggregation behavior leads also in the differences in properties of nanoparticles. It was published several years ago. The neutron neutron diffraction experiment showed different density of the core for gradient and block copolymers. And this different distribution of lipophilic and hydrophilic segments leads also in the different stability and different loading capacity of nanoparticles. Also very important is to find lipophilic part of the nanoparticles, which is very similarly hydrophobic as the drug. You can see here a comparison of solubility parameters of different polyoxazolines and the hypericin, and you can see that the highest proximity and highest similarity you can find in aromatic copolymers of oxazolines, but still difference between solubility parameter of hypericin and polymers. Therefore, we decided to extend lipophilic part in our copolymers, and we prepared gradient copolymers based on ethyloxazoline and phenyloxazoline, these different alkyl chain from ethyl to dodecyl, and gradient character was proved by the kinetic of the copolymerization, where you can see the difference between polymerization rate of ethyloxazoline and phenyloxazoline is about 5 times higher rate of the ethyloxazoline. Means at the beginning of polymerization, we will have polymer chain enhanced on oxazolines, and in next stages, lipophilic comonomer will be polymerized. You can see that differing in composition and different alkyl chain, we can prepare very nice library of copolymers with different thermosensitive properties and different hydrophobicity. For our purposes, the best is the window of thermosensitive properties in physiological range. It means for our polymers between 12 and 20 molar percentage of aromatic moieties. We use these selected gradient copolymers for preparation of nanoparticles. We use dialysis method, and you can see that we are able to provide nanoparticles with size about 20 nanometers, except copolymers with methoxy substituent, and from them you can see aggregation of smaller particles, but lead to enhancement of size of the aggregates. In the after loading of hypericin, the average size was between 10 and 40 nanometers. We studied also stability of a nanoparticle by critical micro concentration values, and you can see that from methyl to dodecyl chain length, the CMC is decreasing. It means stability of nanoparticles are increasing with length of and extension of hydrophobic part. We also measured loading capacity, and you can see when we use 1 milligram per milliliter of hypericin and 4 milligram per milliliter of polymer, we obtain nanoparticles with 15% of drug. Very important is to follow the stability of our nanoparticles, and we studied the distribution of hypericin in different media, and you can see that from methyl to hexyl derivatives, the distribution is gradually decreasing. It means the more stable particles were obtained in the case of the hexyloxyphenyl oxazolate. Analogously, then we followed the uptake of hypericin in the cell. It means the release of hypericin from the nanoparticles. The highest rate was observed in the methoxy derivative, and the lowest was in the case of hexyl-phenyl oxazolate. This side effect affected also photodynamic activity of our nanoparticles. We used two different cancer cell lines, and we followed the viability of cancer cells after photodynamic therapy. You can see in the control there was only light without the hypericin, and no effect on viability was observed. Then we have the pure hypericin and the effect of three different copolymer formulations. You can see that in the case of methoxy and ethyloxy derivatives, with the fast release, the efficiency of photodynamic therapy is very similar to free hypericin, but in the case of ethyloxy, which is most stable, we observed the extended time necessary for efficient photodynamic activity. It means we can regulate the rate of the photodynamic therapy by the structure of used gradient copolymers. At the bottom of slide, you can see MTT assay and viability of cells without light, and you can see encapsulated hypericin did not have any adverse effect on the used cells. For the more detailed study of in vivo behavior of our copolymer formulations, our nanoparticles, we used chorioallantoic membrane model from the Japanese quail embryos, and it is now very popular and efficient animal model to provide very fast and robust information about behavior of your polymeric materials in vivo conditions. The advantage of Japanese quail embryos are leads toward chicken eggs, leads in the very fast development development of the embryos. Already after 8 days, you have a living model for your experiment. In our case, we used this CAM model for testing of fluorescence efficiency. We provided histological analysis and also assessment of vascular damage and angiogenesis expression. Here I will show you a few examples from the results of fluorescence efficiency, image of vessels, and also gene expression of connected with angiogenesis. Here you can see the comparison of the vascular damage for polymers without illumination and after photodynamic therapy, and you can see that our formulation have no effect on the vessels and also capillaries. It means score 1 is no damage. And then when we compared the treatment after irradiation and the effect of photodynamic therapy, you can see that produced oxygen species were able to damage smaller and also capillaries and also smaller vessels. What is important in the treatment of tumor when the nutrition delivery is crucial step in the therapy of the tumors. Similarly, when we assessed gene expression of gene connected with angiogenesis, you can see that hypericin have anti-angiogenic effect, what is beneficiary for the delivery of nutrition for tumor, but after photodynamic therapy, this effect is increasing. When we use our formulation with hypericin, you can see anti-angiogenic effect even after photodynamic therapy, what is very promising results, and can have synergic effect during the therapy. At the end, I will show you our results from utilization of hypericin formulation for photodynamic diagnostics. We used not a laser source of the light, but we used a fluorescence camera, and you can see that when we injected cancer cells in embryos, this is this white arrow, and then we injected our formulation to the embryos. You can see that upon the time and irradiation, hypericin is accumulated in cancer cells, and in such way we can diagnostic place of the tumor upon this aggregation of hypericin. At the end, I would like to summarize my talk, and I hope that I demonstrated that gradient copolymers are very effective materials, able to provide cell assemble and able to provide stable nanoparticles for delivery of different very hydrophobic drugs. We showed that the structure of copolymers and length of alkyl chain can affect the properties of the nanoparticles, and formulations these hexyoxy phenyl moiety extended time of the effect of therapeutic effect, and generally gradient copolymers can be used as materials for delivery of hypericin and used for photodiagnostics and also for photodynamic therapy. I would like to acknowledge help and assistance and cooperation with our colleague in the group of Department for Biomaterials Research in Bratislava, and we cooperate with the University in Košice and Center of Biosciences of Slovak Academy of Sciences, where the experiment on embryo models were provided. This work was also supported financially different national projects. At the end, I would like to thank for your attention.